It is my enormous pleasure and honour to introduce the array of speakers today, and really the collection of talent and the various institutions around the world that would literally not be here today if it were not for Lindon Eaves' efforts and extraordinary talent in developing the field of behavior genetics over the last few years. And so the word Festschrift, not everybody is familiar with a Festschrift and may have sort of looked it up on Wikipedia or something like that. And it's defined as a book honouring a respected person, especially an academic, and presented during his or her lifetime. The book thing, well, we'll talk about that later. It's but for now, at least we get sort of video immortalized presentations. Endel Tulving, a Canadian neuroscientist, proposed that a Festschrift frequently enough also serves as a convenient place in which those who are invited to contribute find a permanent resting place for their otherwise unpublishable or at least difficult to publish papers. This is also such a thing as a web Festschrift, which is compiled by electronic means, has a much greater chance of success given the schedules of the attendees here. And then the other part of this, Festschrift and Lindon Eaves, so Lindon Eaves really defies definition and belies belief. He has 2 CVs. I don't mean the awful car produced by Citroën, but he has 2 curriculum vitae. And it is only one of those that we're gathered today to honour. You may not have found a Wikipedia page for Lindon Eaves. I thought there might be one, there wasn't, so I started one last night, and I encourage everyone here to edit my mistakes out of it and embellish it with your own stories and fun. And finally, a good colleague of mine, Bill Revell, describing at a Festschrift a couple of years ago, said that really it's an answer to the question of how do academics party? Well, basically they get together in a room and give each other talks. And so without further ado, I think we should party, and I'll introduce the first speaker, Nick Martin. So.
All right, well, I don't know about 2 CVs, but I've never been double mic'd before. Anyway, it's my great pleasure to give the first talk here, and I guess I claim right of privilege because I have the immense privilege of being Lyndon's first PhD student. And he, as Mike said, set my course for life, as he has done for many others. This is one of my favourite photographs of him in classic Eaves posture. So some of you may not be familiar with the reference in my title, Paradigm Shifter. When we were getting going in this subject in the 1970s, there was a lot of interest in the philosophy of science and the sort of forward-looking view of our research program versus the degenerative view, as we saw it, of the erstwhile environmentalists. And one of the leaders in this discussion was Thomas Kuhn, this book, The Structure of Scientific Revolutions, who talked about normal science sort of going along and everybody's doing the same old thing, and more and more exceptions would be discovered, so that the model started to drift, and the exceptions became more and more embarrassing to the point where there was a revolution, and then this led to paradigm change or paradigm shift. And I would argue that this is really what took place in the University of Birmingham around 1970. And Lindon was not the only person involved, but certainly played a very crucial role in this revolution. that took place, and really I think is the reason that many of us are here in this subject and in this room. As Mike has already said, Lindon has two curricula vitarum, parallel one in theology, but when I first met him in November 1973, I said, "That's a very unusual combination to be a priest and a geneticist." And he said, "Oh, I think there was another one." But fortunately, Lindon is really not a Savonarola type priest. He's far more Rabelais, as you can see from this photograph taken in Amsterdam a few years ago. So the people who really set the intellectual tone in Birmingham, and Mike Kearsey will talk about this much more, were Kenneth Mather, and who worked on Drosophila, and his student John Jinks. Mather was a student of Fisher's, and John Jinks was Mather's student, and Eaves was John Jinks' student, so direct line of descent. And I'm Lindon's student, so I guess I'm fourth or fifth degree separated from Fisher, which is kind of nice. It was really the experiments done on this plant, Nicotiana rustica, in this huge field out the back of the genetics department in Birmingham that really set the standard for, these experiments consisted of up to 10,000 plants, giving enormous power to dissect out genetic components using very clever algebra for biometrical genetics that Mather and Jinks extended from the earlier work of Fisher. But it was what really got all of us engaged in this subject, I think, was this paper that was really a watershed in the history of the subject. This paper by Jinks and Fulker, published in 1970. This is a very nice portrait of David Fulker, painted by Constance Holden, who sadly was killed by a security vehicle in a Washington parade a couple of years ago. But she was as well as being a journalist on science, she was an amateur painter, and I think she's captured David very nicely there. Incidentally, just a little plug, this website here, you can find all the papers I'm talking about today and many others. I've sort of tried to, over the years, accumulate a lot of the key papers in the subject as I see it, and I welcome suggestions for others, particularly if you send me a PDF, I'll post it. But this paper, which set out at great length to expound this new view of the subject, saying, look, the whole subject is beset with these little itsy-bitsy studies, many of them on a couple of dozen pairs of twins or very small numbers of relatives, very sort of post hoc analyses, just simply looking at correlations and saying, oh, well, it looks genetic, or it might not look genetic, and absolutely no rigor about it. And in this paper, they expounded a systematic way of looking at these data and setting out to examine things systematically. And Lindon was right in the middle of this, and I just would like to list what I see as his main contributions. The list is not exhaustive because there's as many dots as I could fit on a page, but as I said, the main heuristic, I think, was rigorous hypothesis testing of whatever your ideas were, and contributing to that, the design, what relatives do we need in our sample, the power. It's all very well, having these nice designs, but how big do the samples have to be to have any hope of detecting the effects that we think are there? And what are the mechanisms? I mean, it's kind of boring if all you ever do is estimate heritability. What is the role of assortative mating? How important is genetic non-additivity? Are the same genes operating in males and females? Do relatives actually, the genotypes of one relative actually influence the phenotype of another relative? When we're looking at a large number of, say, IQ tests, or subtests, is it the same genes influencing all of the tests, or are there some genes in common and some genes different? And sort of taking that to the particular case of longitudinal development, how do genes switch on and off during development? All of these are topics that Lindon, I think, did the pioneering work on, and I'll really not cover them in any detail. Others during the day will do that. I just had to put this up just to show that, in fact, Lindon was already well in this game before Jinks and Fulker. This was his first paper in 1969, and already showing his concern with design, what designs in terms of relatives that you would use in your experiment would actually enable you to disentangle different components of variance. And here's another one in 1972 extending that theme. But in between these theoretical pieces, he also had time to join battle in some of the great, what were blatantly ideological plays, including this one from Sandra Scarr and Late of This Parish, and who published this sort of landmark paper in 1971, which was really a great idea, showing that there were differences in heritability between different racial, social class groups and sort of prima facie evidence of discrimination. Only trouble was that Lindon and John Jinks showed that the power of their study and the way that they'd done it just wasn't there at all. And so Lindon was never shy of joining a good fight. Very early at this time, I should say, I mean, I'm just talking about the 1970s, which is the time when Lindon and I overlapped most closely, well, at least initially. And what an astonishingly productive decade that was. As you'll see, many of the themes that many of us have been working on since that time really were adumbrated in Lindon's work. This is a paper in Nature in 1974 with the first analysis of the genetics of social attitudes. Completely indigestible because, of course, nobody thought about standardising variance components then. They just gave you the variance components on the raw scale, which was pretty terrible, looking at the heritability of radicalism and tough-mindedness. As I said, Lindon was the first to actually consider, at least in the human context, what the effect of one relative living with a relative of a different genotype, or if they're an MZ twin of the same genotype, would have on their own behaviour, introducing the concepts of gene-based competition and cooperation. And I think probably Greg Carey will be talking more about this in his talk. He extended this to a little bit later. Lindon was the first to think about genotype by age interaction, the fact that different genes can be affecting the same trait at different ages. And how would you pick this up? How would you go about picking this up? And this very modest little contribution to behaviour genetics of a couple of pages, he sort of provides the first analysis of this. Once again, something that many of us have spent the last 3 decades keeping on working on. Are the same genes influencing males and females? When you're doing twin studies, one of the traditional things that people did was to throw out opposite-sex twins because you wanted to control for sex. Of course, what they're doing is throwing out the very information which would enable you to see whether the same genes or indeed environments are operating in males and females. You can think of that sort of verbally like that, but Lindon was the person who actually set up the algebra and quantified how one would go about doing that. And of course, we've now got countless examples of sex limitation. And I think the thing I'm most proud of is the work that we did together looking at genetics of covariance structure, which really, I think, got multivariate genetics on the map. But one of the points, the main point I want to make here, is not to trumpet myself, but just to make the point of how incredibly generous Lindon has always been with authorship. Basically, I was just the amanuensis, all the ideas were his, and I was just the gadfly who kept nagging and nagging and nagging to make sure that this damn thing got out. The same with power studies and sort of getting, because Lindon had done wonderful power studies for designs that nobody ever used and that were actually impossible to ever gather. And I said, well, what about, just the plain old twin study, which is what most of us have to do. And that was really what stimulated this first set of power calculations, which I think Neale will tell us were actually wrong. But anyway, at least it set out the principle. And the most important consequence, incidentally, we're very pleased that Mike Kearsey was a co-author on this paper, and Mike will be speaking later about Birmingham days. But the main upshot of this, at least for me, was to suddenly look at these look at these numbers of pairs that you need down here to sort out some of these quite common situations. Huge numbers, much bigger than anyone had been using in any twin studies to date, and it really underlined the need for huge twin registries, and that was what stimulated me to go back to Australia and start the Australian Twin Register and recruit the first 15,000 pairs, and I think was a stimulus in the foundation and expansion of other registries like the Dutch Twin Registry and expansion of yet other registries. Basic message, you can never have enough twins. And if you look at the Scandinavian cancer twin study now, they are up over, I think they've collectively got nearly 500,000 pairs or something like that in the Scandinavian cancer registry. So all good things have to come to an end, and Lindon left Birmingham in 1979, moved to Oxford and was at Lady Margaret Hall. And I don't think I'd be far wrong in saying that perhaps the most important upshot of his time in Oxford was actually the recruitment to the subject of Andrew Heath, who did his PhD DPhil, in Oxford under Lindon, and subsequently moved with Lindon. Well, soon after Lindon got to Medical College of Virginia, moved there, as did I, and as did Ken Kendler. And so from 1983, we had a terrific little scene going there at MCV in Richmond, and another fairly astonishingly productive time. He also managed to find time to fulfil his other part of his CV in marrying me. And when it subsequently showed him this photograph, I said, "Oh, I must have washed my hair that day." This was the time when we were able to consolidate the work that over the years Lindon and Hans Eysenck and I'd made some small contribution to on personality, the genetics and cultural influences on personality and behaviour. And I really wanted to just include this slide to acknowledge the enormously important role that Hans Eysenck played in the intellectual development of Lindon and of myself, and in the early days of the field, much missed. Likewise, in Social Attitudes, this is a paper we published in 1986 coming out of that work, analysing data on about 4,000 pairs of twins I'd collected in Australia. A paper that, as Lindon said, disappeared like a stone down a well, totally ignored. Nobody paid any attention to it at all. Until about 25 years later, a group of political scientists in Nebraska, including Peter Teamy, who's present and will be speaking later, actually rediscovered this work, and now it seems to be sort of quite popular in political science. But this paper, I think the thing that we're most proud of in this paper is Lindon and I agonised for weeks about how we would end this paper. And I mean, because everybody knows that social attitudes are determined by the environment. You know, how could genes possibly take part? And after a long time, I think we encapsulated reasonably well. In no way does our model minimise the role of learning and social interaction and behavioural development. Rather, it sees humans as exploring organisms whose innate abilities and predispositions help them select what is relevant and adaptive from the range of opportunities and stimuli presented by the environment. The effects of mobility and learning therefore augment rather than eradicate the effects of the genotype on behaviour. And fine words, I think we wouldn't resolve from those now. The other great enterprise that took part in the 80s at MCV was really extended from work that Lindon and Andrew had done on, well, supposing we go beyond twins, you know, how much more could we dissect of the particular mechanisms of variation? So by adding parents, siblings, spouses, and offspring to twins in the extended twin design, in fact, you generate a huge number of correlations, I think 80 correlations taking account of sex, and can estimate a very large number of parameters. In fact, one of the very first applications of this was to a phenotype dear to Lindon's heart, church attendance, where indeed we showed both in Australia and in the US that there are, certainly in males, there are genetic effects up to 22% in Australia, 27% in the US, for males and 15 and 19% for females, influencing church attendance. And so there have been many other analyses around this, but of course these Festschrifts are always held towards the end of someone's career, often when people have retired or thinking about retiring. But I just want to suggest to Lindon that he can't possibly retire until he's written Lindon's last paper, which is Finding the God Gene. And we just need to go out there and collect this very large sample of priests and nuns, and then get a bunch of Australians as controls, being the most irreligious race on earth, and do the GWAS for God. Sorry? Yeah, exactly. This is what we work on. But anyway, let us give thanks for Lindon's life and career, and I just end with the fact that you need to actually watch your paradigm as things go on. Thank you, Lindon.
So a Festschrift has a dual function. It is both a celebration of the scholarship of a wonderful scientist and thinker, and it is an opportunity to challenge us to take the ideas of that scholar and continue to run with them, and to continue the activities of the next generation and the next generation after that in the Eaves pedigree. I would love us to draw up the Eaves pedigree. So I'm an Eaves intellectual child. Mary Waldron is a grandchild. But we must be in the great-great-great-grandchildren generations in some parts now. So I'm going to talk about mate selection, arguably the second most important question in evolutionary psychology. Critical for determining what happens to the genes that we pass on to our offspring, which genes are passed on, the quality of the environment that's transmitted to our offspring. From the behavioral economic perspective, a real puzzle. Why are we so bad at it? What are we doing? How are we selecting our partners? How are we deselecting our partners, which seems to be so common these days? We're doing without partners. When are we reproducing? And Lindon very early on saw that understanding the human mating system was going to be critical for understanding environmental influence, resolving cultural and biological inheritance. For things as simple as the shared environments in twin data. Are there really shared environmental effects on social attitudes, or is it really just the genetical consequences of assortative mating, as that great PNAS paper that Nick quoted suggested? Lindon was the first to think about assortative mating as a multivariate system, and therefore a system in which we could see asymmetries by gender. And to see that mate selection raises fundamental evolutionary questions, although I think questions that we've never been able to make much progress with. Lindon has always been great at linking back to the past to see the work by the pioneers in the field, in this case, Sir Ronald Fisher, and at the same time trying to come up with new strategies for understanding what's happening. In this case, thinking about, let's start with a twin design, let's move out and look at the spouses, the partners of these twins to analyze the human mating system. Early Eaves omissions. One thing you should know about Lindon, he has been very happily married to a woman he has known since a very early age. A consequence of that is that Lindon never paid much attention to the questions of divorce or separation from a reproductive partner, timing of reproduction, infertility, infidelity. It fascinates me. I love the story of the male fruit fly who would try to mate with a blob of cotton wool on the end of a cocktail stick. Because all around him, he had colleagues who roughly approximated the male fruit fly in their exploratory behaviors. And yet Lindon had this dedication to his partner that influenced his science. He never got to thinking about how mate selection and mate deselection had to be considered as parallel processes. I like to think about human mate selection as an unfunded scientific mandate. A scientific mandate because it's so critical to the questions that we like to think about, and an unfunded scientific mandate because you sure can't persuade NIH staffers that this is significant from their perspective. So I draw it again to your attention because when your romantic young PhD students are looking for topics, hey, this is all about romance. So the challenges of characterizing human mate selection, obtaining data that adequately characterize what's happening, and finding models that approximately characterize what's happening. And I've given us all a failing grade. I don't think we're there yet. And that's why I thought this is ideal for this Festschrift. Lindon started the ball rolling, but we've got a long way to go. And the challenges for this one are pretty serious. One is reciprocal influences. People influence one another as they live together. And yet, how do we do prospective research? How do we get away from the recall biases, all the complexities that happen in real data? And then deselection. I don't care about partnerships that break up when there have never been any kids, except insofar as that causes a delay in reproduction. But it does become important when we think about the outcomes of reproductive partnerships. Timing of reproduction becomes important. Sampling bias becomes horrendous when we allow for human subjects' autonomy. If I start with a reasonably high response rate, and now I'm looking at 4 individuals, my sampling biases have huge potential for distorting what I'm seeing in data. And so the data that we actually observe, if we do a conventional cross-sectional study, and I speak as one who has tried such a study, studying twin pairs and their spouses, we have all these processes superimposed: selection, deselection, cohabitation, recall biases, as we try to reconstruct what's happening. A nice concrete example, I work with female alcoholics as part of my research program. Delayed marriage, delayed reproduction, accelerated breakdown of relationship, abusive relationships that mean you can't talk to the partners, accelerated mortality. So when we look in the real world of data, we face some challenges. Okay. Can the traditional behavior genetic sampling strategies help us in this area? Volunteer panels, I call it Hail Mary sampling. Because you've got to pray that your biases are not going to give you completely misleading results. Cohort studies in systematically ascertained populations still a challenge because of all the effects we've just been talking about. Small population studies, well, do we really want to limit our knowledge to the population of Iceland? So I've gone back to thinking about some traditional epidemiologic strategies. Ecological studies in epidemiology, wonderful ways of getting false positive findings. Analyzing group level variables, these could be studies of local cancer rates and poverty, international comparisons. Creating what the epidemiologists call ecological bias. because you're not measuring individual level variables. And a solution that the epidemiologists have used is the so-called cohort ecological study. You combine population level data with cohort studies. And by doing that, you work around many of the disadvantages of the traditional ecological study. If you think about it, many of us, when we do cohort studies, are actually starting from population data. And we're selecting out a sample. And we're discarding the rest of that population. But that stage of discarding the rest of the population, we don't have to do. And so as we start to try and think about getting rigorous data about assortative mating mate selection, keeping the rest of the population in our analytic frame becomes an important strategy. So perhaps combine state-level databases with cohort studies, targeted sampling based on a fill-in-some gaps strategy. So a thought experiment. Let's think about educational level, height and weight. We can take birth record data and we can identify birth sibship strategies, cousinships. We can identify reproductive partnerships. We know from the presence of a child that this partnership reproduced. We don't really care about the non-reproductive partnerships anyway. And we can do the experiment and say take height and weight data from, in the United States, driver's license records. I actually did the link to cohort data, and amazingly, you do about as well extracting an individual's height and weight from what they report on their driver's license when they're young to what you get if you ask them for those information in an interview. So in this case, good validity. And we can pull educational levels from the birth record. There are some challenges, immigrants and emigrants, but our cohort studies will tell us about that. Incomplete educational levels at the time of a birth, but our cohorts will tell us about that. Unmeasured individual level predictors. From our cohort studies, even though our cohort participants may never intermarry, still having the prospective data will allow us to test some critical assumptions that we're not testing when we rely on retrospective data. Quality control issues for state databases, those are significant. And again, you need the cohort data to tell you what works, what doesn't work. Valerie Nopick in this audience will tell you how if you identify a series of mothers who've had apparently one pregnancy where they smoked and one pregnancy where they haven't smoked, 75% of those are false positive. But it's still a very efficient strategy for getting the other 25 that are true positive. And suddenly we're going to start running into big data problems because now you're talking about working on a scale of databases of millions of individuals, which when you start to try and match and merge does become a headache. And you're back in the business of Bayesian analysis. Because for many of these cohort ecological problems, you're back to Bayesian modeling. And so the good news is we've been doing this anyway, building a population and family register for an unnamed Midwestern state. The bad news is just to get all the approvals to do a secondary analysis of the data you already have, you're probably going to spend 12 or 18 months getting all the sign-offs. So I can't illustrate with real data. So I'll move on to some actual data because Lindon never limited himself to thinking through the issues. He also wanted us to work with data and use the data to refine our modeling. and begin at the end with human mate deselection, separation and divorce. The persist or quit problem, I like to call it. Anonymous, an anonymous state adolescent female twin study, multiple waves of data starting at median age 15, high quality sampling, relatively few families lost to the study. About 2/3 of the families grew up with both biological parents. Don't forget that the 50% divorce rate that you always hear about, 50% of couples, not 50% of reproductive partnerships. Of course, a much higher separation rate for African American families. Just some quick illustrative numbers. Look at the far most rightmost column. This is the concentration of early onset adolescent alcohol, tobacco, illicit drug use, early meaning before age 15, in families that are separated. That means never married, divorced, any sort of relationship separation. So about 30% we'd expect if there was no effect. It's high. Control for things like parental alcoholism doesn't change very much. Early sex, early depression, early suicide attempt, concentrated. If you want to efficiently find these high-risk kids, starting with separated families is a very efficient way to go. And by the way, if you want the kids who've been raped or molested, it's also a very efficient way to go. And that one's true also in African American families. So the behavioral economics problem. You've got a lousy partner, do you persist, do you quit, or do you quit and try again? How do we overcome the strong confounding between parental separation and parental psychopathology? Of course, ideally, what we want to do is to randomize alcoholics to separated versus intact relationships. And I thought about that and I thought, aha, Australia. And I say that because Australia lagged behind America in the changes in its divorce laws. Britain too lagged behind, but they happen to have Australian data. This is just a cohort of twins. Notice the shift in the rates of parental separation purely as a function of when a person was born, because this is a period of transition for the uptake of parental separation. Prior to these birth years, divorce in Australia was on the grounds of abandonment, cruelty, proven adultery, and so very uncommon. A sort of mating for educational level looks the same in intact families and separated families. Very weak correlation between educational level and separation. Significant, it would get some sociologists really, really excited, and actually positive in those days. So the more educated were more likely to try out this innovation of divorce. That's completely reversed now, of course. These are data on alcohol dependence, comparing intact families with no paternal alcoholism with paternal alcoholism, families where parental separation has occurred with no paternal alcoholism and with paternal alcoholism. And the question is, if dad's alcoholic, the kids do better if he leaves or if he stays. Under genetic confounding, what we'd expect is the worst risk would be when dad leaves, because that's where the highest genetic risk is concentrated, the most severe alcoholics. But for this cohort in transition, the families where dad gets kicked out, the kids do better. kids do better. Quitting looks like a better strategy until you look at depression in the daughters. Because the families where the alcoholic dad gets kicked out, the daughters do much worse. And unfortunately, the reason they do much worse, one of the reasons is there's an excess of molestation, rape, other trauma in those daughters. So we don't truly yet have randomization, but we have data where we can do more elaborate matching to try and better do the experiment. So I would say we're far from having a complete understanding of how best to model human mate selection and deselection to better characterize the transmission of risk to the next generation. I have to thank Lindon for helping us lead the way in thinking about this. And Sue, I forgive you for being such a faithful partner that Lindon forgot about the deselection part of that puzzle. And I'd like to suggest that when we work with these population databases, sometimes you want to preserve the entire population database and still work with the cohort studies within that population database. And finally, as a medical school employee, I have to announce the absence of any corrupting influences from big pharma and other sources. Thank you.
Anyway, it's a great pleasure to be here. It's coincidental also, I think it's coincidental, this International Quantitative Genetics Congress here in Edinburgh the same week. It's the last day today. I sneaked out for this, so you'll have to forgive me if I sneak out again at the end of this and go back to my other meeting. I was around when Lindon was doing his undergraduate and his postgraduate work at Birmingham, hence I think this is probably why I was invited to come along and make a few comments about the early days. So I start with a little bit of the sort of early history. Nick's already shown a picture of the University of Birmingham. This is it. It's for those of you who are not from this country, it's right in the middle of the UK. It's sort of a dead centre of the place. Can't get much further from the coast. And I'm going to say a little bit about how the genetics and quantitative genetics started in Birmingham. As Nick has said earlier, Mather was the first person to get it started. He came as a professor at the University of Birmingham, the first professor in genetics they had in 48. And he stayed as professor until 65 there, where he moved off to Southampton as a vice-chancellor and then eventually got his knighthood. As Nick had already said, was a student of Fisher, and many students of Fisher, as I'm sure you will know, spent most of their life trying to explain what Fisher had done to other people. Those who're not familiar with Fisher's teaching, he would put some formula on the board and says, right, we'll start this here, and it's fairly obvious, isn't it, that it goes to that. And people would say, well, I can't appear to be stupid. Of course, it has to be obvious, but they would spend the next 30 years trying to understand that small step that was so intuitive to Fisher. Mather did a lot of this. He was also very interested in cytogenetics. He was interested in statistics. He wrote a very important book on linkage, which set the scene for the methods that have been used over the last 20 years in terms of major linkage studies. But the general principles were set out by this book. Mather also introduced the concept of polygenes. I think the term polygenes was a Matherian invention. It's coming back into phase again now, but of course QTL superseded it later, but Mather started the whole idea of polygenes. And he did a lot of work using Drosophila, trying to locate and find out how many genes there were controlling quantitative characters. Now, of course, this was in the days before molecular markers were available, so he had to use major gene markers, and he was working in Drosophila, for which there were a lot of markers available. And he was able to show a very important thing, that no matter where you looked for genes controlling a quantitative trait, you found them. And Nick reminded me last night that his successor, John Jinks, who I'll say something about in a moment, was once asked how many genes are there controlling a quantitative trait, and he said it depends how much work you put in looking for them. The more effort you make, the more you will find, and this of course is what we know from GWAS studies now, that everywhere people look, they find them. So Mather started the idea of looking for polygenes by searching chromosomes in Drosophila. This work was taken up in Cambridge by Foley subsequently. John Jinks was Mather's student, and he got his PhD in 1952. A Birmingham lad, he did his undergraduate at Birmingham, carried on, did his PhD in Birmingham, and I think he would never have left Birmingham if there weren't trains and things to take him to London occasionally. But Birmingham was the centre of his universe, and he couldn't understand why anybody would want to go anywhere else. And in fact, I think as Lindon discovered, if someone does leave, you've lost it completely with John Jinks. He wasn't very happy at all. What's wrong with his city? John Jinks was very interested in various things, but biometrical genetics, quantitative genetics probably was his main interest. He was also did some seminal work on cytoplasmic inheritance. He got interested in fungal genetics. So, like Mather, fairly broad interests in genetics, but biometrical genetics was his key. Plant biometrical genetics, he's working with tobacco, and Nick Martin showed some images just now crossing tobacco plants, running very, very large experiments. Mather was so Jinks was very interested in getting quantitative genetics applied, particularly applied to plant breeding, but also animal breeding, and this was very important in getting the behavioral genetics off the ground in the late or early 60s. He had senior management roles in the university, but also in a lot of the bioscience institutes, and therefore he was able to sell the importance of genetics to the nation generally, and also to the wider world. The next critical person, I think, was Peter Broadhurst, who was at the Maudsley Hospital. He had worked closely with Hans Eysenck, and he came to Birmingham in the early 60s. He'd written a very seminal paper in the late 50s in which he'd actually referred to Jinks and Mather and biometrical genetics, and how this approach could be used for behavioral studies. You will all know, of course, that Broadhurst was instrumental in developing the Maudsley strains of rats for response to stress, and when he was there at the Bethlehem Hospital. So he came to Birmingham, and there we had the critical group there of John Jinks, Peter Broadhurst, who met regularly and tried to, I think, get the whole subject of quantitative genetics for behavior up and running off the ground. Now, there are some other connections with Birmingham people, and you may not recognize this fact, but both of these guys, Galton and Cyril Burt, are lads from very close to Birmingham. Francis Galton and Cyril Burt both lived in adjacent villages just south of Birmingham, and my wife and I went out for a Sunday meal quite recently in one of these villages, and wandering through the churchyard there found Francis Galton's grave, fenced off with quite a high barricade, very simple grave. Here lies Francis Galton. Burt was brought up in the village next door. Galton was in Claverton, Burt in Snitterfield, very close together. Didn't know each other, although Burt's father knew of Galton and knew of his work, and he got interested in psychology that way. So this little bit of background is to say by the 1960s, the environment was right, really, to get going with behavior genetics at Birmingham. In 1964, John Jinks started a master's degree course at Birmingham in applied genetics, and one of the first graduates from that course was David Forker. David graduated in 1965, only 3 students on the course. Couldn't teach courses for 3 students now. Wouldn't be allowed, wouldn't be economically possible. Universities wouldn't allow it. So had David Forker been around in this time of the year, it would never have got going. So David Forker got his BS, he got his master's degree there, having got a degree in psychology earlier at Birkbeck. And in 1966, as a result of his master's degree research, he published a very influential paper with John Jinks on rat genetics, which was a very important step forward in this area. In 1966, we have Bert's classic paper, The Genetic Determination of Differences in Intelligence, a study of monozygotic twins raised together and apart, where we have this heritability of 0.77 that came out then. This wasn't of any great surprise, I think, to people at Birmingham. They knew very well that for all other characters that they looked at in plants and animals, heritabilities of sort of 50 to 70% were quite normal. 77% didn't raise any eyebrows at all, I think, at that time. But clearly it did in other circles. As you are all aware, the authenticity of this data was subsequently denigrated came in by Gilly, by the Clarks, and the whole of Bert's work and contributions were very, very seriously damaged, particularly by Hernshaw's biography of Cyril Burt. The consequences of this, of course, subsequently for the whole area of quantitative genetics in behavior were very serious. So we get up to 1966 with Bert's paper, and here comes Lindon, and I've stolen this from Nick. Lindon got his BSc in 1966, and we were delighted that he, because he did brilliantly well, he'd done almost the same course that David Forker had, and John Jinks thought, well, here was a wonderful opportunity to have the 2 of them there together in the department. Come on, Lindon, you're going to do a PhD. No, he decided to push off and go to some other university a little bit further south, called Oxford, I think it was, to do divinity. And we will wonder whether we'd ever get him back, but of course a year later he did come back, armed with the special feature that we'd always lacked in this area, and that was divine revelation, which did us no harm, or did them no harm over the future years. Lindon carried on to do his PhD, and I think got his PhD about 1970. David Forker had got his the year before. So with the start of the psychology department there, Jinks, Broadhurst, now Eaves, Forker, and links with Hans Eysenck, there was fantastic opportunities to get this work off the ground. And the rest, some might say, was history. In 1969, I don't know whether people might recognize themselves in this picture, they held a NATO Advanced Studies Institute. Jinks and Broadhurst is here. I'm hiding away over here, so I had a very vague connection with behavior genetics in those days. This guy's David Hay, I think is still active. Is he here at the meeting? In Australia, Twin Studies. There's David and Lindon. Slightly younger version there, but in the final year, I think, of his PhD. Here is a picture somewhat later This is in 78. Lindon and Nick Martin here. Still? I think he's got the same jacket on, I can't tell. A few other people. You talked last night, Lindon and Nick, about Jeff Gale. Here he is in typical pose, I think. So this was the genetics department in 78, just before you left and went to Virginia. Nick has said an awful lot about your contribution in terms of what you were working on and what you were studying at that time. And what was very important, I think, to all of us at that time was that you were actually looking at the power of different designs, how the relative sizes of different types of family and twins, contribution of twins raised together and apart, all of these factors, how big the study should be, what sort of power you had for distinguishing different types of genetic variants, played a very, very important part. And I think by the end of your period at Birmingham, I think you'd written about at least 30 odd papers, and on the basis of this, you were awarded your DSc at Birmingham. One other thing that happened at that time, which I think both Lindon and Nick were very instrumental in, was starting a twin registry in Birmingham. It took quite a long time to get this going, and Birmingham Education Authority were very helpful and collaborative in this. As I recall it, it was a matter of getting the Education Authority to identify children who had the same surname and happened to be in the same class at school, and therefore were likely to be twins. And then they were contacted by the Education Authority if they were prepared to collaborate. Then Lindon and I think Nick too were involved in trying to get them genotyped and then try and get them into the registry. I don't know very much about what came of the data, not a lot, no. But in the late 70s, of course, Lindon was to leave. Broadhurst left and was very seriously ill quite soon after that. Nick had gone back to, had left as well. John Jinks died quite soon after that. And in the 80s, it was very, very difficult to get funding for behavioral research, to try and maintain a twin registry, particularly when there were no behavioral geneticists left, was very hard. We struggled for a long time to do this because we needed resources to keep up to date, to contact people, and of course, once you've lost contact with these twins, it was very difficult to make any use of them. And the whole issue about behavior genetics and its, there was fear of anybody being involved in this area. Isink and many others were, lives were made very, very difficult and treated almost as fascists. So it was very, very hard at that time, and funding agencies had limited money. They wanted to put it in biotechnology and more popular areas, and behavior genetics wasn't top of their list in the UK at that time. So in the end, we had to abandon it. So sometime in the 1990s, the whole thing was destroyed, and it's a shame because a lot of effort and a lot of your time went into getting this off the ground. There is another one now, I think there was a twin registry in Birmingham, but it's run by the medics. Right, well, let me just sort of finally just say something about Lindon's interactions at Birmingham. It was a fantastically interesting time when these guys were there. There was constant discussion over tea and coffee and beers and whatever on all subjects to do with statistics and behavior, genetics, design of experiments, and so on. Lindon's religious views were a subject of great interest. The genetics department at Birmingham was rather like Nick Martin's description of Australia, a group of atheists. I think that Lindon was the only exception. And when we tried endlessly to try and find out what it was that it kept Lindon with his faith, almost everything we went for, he said, oh no, I don't believe that. No. What about this? Oh no, nothing but, So almost all the main sort of tenets of the Church of England, Lindon didn't seem to believe in any of them. So we'd ended up, we thought, well, probably it was the incense and the communion wine and the theatre of the whole thing. The one other important contribution that I would like to acknowledge here is that one of the interesting characters that plant geneticists were looking at and regularly looked at was flowering time. The flowering time is an important trait. It's a measure of how plants develop. That's the time from sowing the seed to when the flower opens or the seed is set. Flowering time, very, very important trait. Nick got quite interested in this, if I remember, and he showed a picture there of somebody actually doing some crossing with open flowers. And he said, well, we can't really study flowering time in humans, but how about deflowering time? Wouldn't that be something that we could look at? And Nick and Lindon spent some time discussing this possibility. And if I can just jump ahead here. I'll come back to this one. I was pleased to hear that, in 1977, they actually put this into operation. With icing, they had a paper essentially looking at one aspect of deflowering time, and Nick, I think, has also published 3 others or something in this general area as well. I'll just go back to the previous slide. This is a meeting of the Genetical Society in 1987, I believe. And there's David and Lindon just coincidentally standing very, very close to each other at that meeting. There may be a few other people that you can recognize there, here. From people from Edinburgh, Bill Hill's up here. Lurking just to my left here is Chris Haley, who's organised the International Quantitative Genetics Congress with Bill Hill, but was also instrumental in seeing some of the final work in behaviour genetics at Birmingham. Lindon was always a bit of an extrovert. We had wonderful parties. Sometimes we remember him having parties at our house, sitting on the swing, singing quite risque songs, if I remember, full of sort of colourful language, quite loud at night after a few drinks. I have identical twins myself, and they still recall these with interest. So I'd like to finish by congratulating Lindon on his very great achievements over these last 50 years. Extremely productive career, and I'm quite sure this productivity will continue in the future. I feel safe in the present company to say that this is probably almost entirely due to his genes. But I would like to suggest that perhaps Birmingham had a small environmental effect on him in the early stages. Lindon, I wish you all well, and thank you very much for inviting me to talk.